Next comes stages of embryonic development. It includes cleavage, morula, blastulation or blastocyst formation, implantation and gastrulation. First of all, cleavage. As already said that cleavage is influenced by yolk, which being a non-living and inert material retards the cleavage furrow passing through the cytoplasm. So cleavage is a successive mitotic cell division of the egg, and in human zygote it occurs during its passage through the fallopian tube to the uterus as in other mammals. So cleavage is holoplastic, and first cleavage is completed after 30 hours of fertilization. Cleavage furrow passes from animal vegetal axis as well as center of zygote. It divides the zygote into two blastomeres and it is slightly unequal. Thereafter, the rate and pattern of cleavage is non-specific. Next is morula. See, cleavage results in solid ball of cells called morula. This is the solid ball of blastomere. Phase of compaction ensues in 8 cell stage. Morula has almost the same size as that of fertilized egg due to presence of zona pellucida. Morula has 16 to 32 cells. Zona pellucida is still forming the outer cover, and morula undergoes compaction. The outer peripheral cells are smaller or flat with tight junction, while the inner cell mass slightly large, rounded with gap junction. See, the cells are compacted and of two types: outer slightly smaller peripheral cells with tight junctions, then the inner mass of cells with gap junctions. Morula descends slowly towards uterus in 4 to 6 days. So during cleavage, the young embryo descends in the fallopian tube slowly due to feeble fluid produced by epithelial secretion and cilia. Ultimately, it reaches uterus. It takes 4 to 6 days, and corona radiata dissolves away during this period. So corona radiata detaches during this period. Next is blastulation. Morula form trophoblast or trophoectoderm. See, in uterus, the endometrial cells becomes full of nutrients which are also secreted into uterine cavity. The same is called uterine milk. As the young embryo reaches uterus, its outer cells begin to absorb nourishment and grow while covered by zona pellucida. Consequently, the outer cells enlarge, flatten and form trophoblast or trophoectoderm. So morula form trophoblast or trophoectoderm. Trophoblast secretes a fluid into the interior. So trophoblast pours a fluid towards interior, producing a cavity called blastocoele or blastocyst cavity, and it creates a cavity called blastocoele. With the formation of blastocoele, morula is converted into blastula, which is called blastocyst in mammals. So the embryo is now called blastocyst. Blastula contains a fluid-filled cavity called blastocoele surrounded by one or more layers of cells, the blastomeres. So it is equivalent to blastula of other animals. The size of blastocyst is roughly 3 times the size of morula. So blastocyst stage is completed after about 5 days of fertilization. Trophoblast later becomes 2-layered outer syncytotrophoblast and inner cytotrophoblast. It secretes human chorionic gonadotropin, forms villi for implantation, and later on produces chorion, amnion and fetal part of placenta. So blastula with blastocoele is also called coeloblastula, like in frog. You can see here the development of blastocyst. This is 2-cell stage, this is 4-cell stage, 8-cell stage, and this is morula. Morula is converted into blastocyst. This is less blastocyst. So this is the development of blastocyst. Next is implantation. See, it is embedding of the blastocyst into endometrium. So it begins in blastocyst stage after about a week of fertilization. Blastocyst creates a slit in zona pellucida partly due to its growth and partly by secreting trypsin-like enzyme. The surface of trophoblast secrete lytic enzyme which cause corrosion of endometrial lining. They also give rise to finger-like outgrowth called villi. See, trophoblast cells secrete lytic enzyme, cause breakdown of some endometrial cells and absorb nourishment and divide themselves to form villi. Villi not only help in fixation but also absorption of the nourishment. So villi penetrate endometrium for fixation and absorption of nourishment. So implantation causes nutrient enrichment, enlargement of cells and formation of uterine part of placenta called decidua. So the attachment of young embryo blastocyst to endometrium of uterus is called implantation, and implantation requires 3 to 4 days. It is completed by 9 to 10 days after fertilization. So implantation brings about nutrient enrichment and further enlargement of cells of endometrium or mucosal lining, which is now called decidua. Now decidua has 3 regions, or decidua differentiates into 3 regions. First one is decidua basalis. It is a part of decidua underlying the chorionic villi and overlying the myometrium. Second is decidua capsularis. It lies between embryo and lumen of uterus. And third is decidua parietalis. It is part of decidua that lines the uterus at a place other than the site of attachment of embryo. Now diagram of implanted blastocyst. See here, this is corpus luteum maintenance, regulation of cells, invasion of gene expression. This cytotrophoblast, this is endometrial glands, these are growth factors, this is endometrial capillaries, endometrial stroma, this is hormone progesterone, helps in the regulation of endometrial function and trophoblastic proteolytic activity. This is implanting blastocyst. And cytokines and growth factors regulates HCG, HPL secretion, proteinase activity and trophoblast invasion, trophoblast proliferation and differentiation, and endometrial proliferation, secretion and decidualization. This is all about the implanted blastocyst. Next is C. First there is a formation of secondary oocyte, fertilized ovum. Here fusion of pronuclei produces a zygote. Zygote is further going to cleavage, which forms morula. Morula differentiated into blastocyst. Blastocyst is ultimately implanted in the endometrium of the uterus. Now gastrulation. See, it is characterized by movement of cells in small masses or sheets so as to form primary germinal layers. Gastrulation is characterized by cell movements or morphogenetic movements that establish the germinal layers and initiate morphogenesis. There are 3 primary germinal layers: endoderm, ectoderm and mesoderm. So endoderm is the first germinal layer that appears in human embryo. Next, the cell movements that occur during gastrulation are called morphogenetic movements, and the product of gastrulation is called gastrula. You can see here the gastrulation. This is trophoblast, this is endoderm, amnion, this is extraembryonic coelom, amniotic cavity, allantois, this is placenta, allantochorion, this is extraembryonic coelom. See, in gastrulation there is a formation of endoderm, there is a formation of embryonic disc. After formation of endoderm, the remaining cells of inner cells mass undergo regular arrangement and called embryonic disc. Third, formation of ectoderm, that is the embryonic disc grows to produce a complete layer below the trophoblast. It is called ectoderm. Amniotic cavity. Amniotic cavity is a space develops between ectoderm and trophoblast. Trophoblast adds special cells to its roof. They are called amniogenic cells. The cavity is called the amniotic cavity. It is filled with fluid-filled amniotic fluid. Formation of extraembryonic mesoderm is also a part of gastrulation. Here cells from trophoblast proliferate between endoderm and trophoblast at the abembryonic pole and between trophoblast and amnion at the embryonic or animal pole, called extraembryonic mesoderm. And lastly, there is a formation of intraembryonic mesoderm, in which cells of embryonic ectoderm proliferate at one end to form a ridge projecting into amniotic cavity. It is called primitive streak. This is all about gastrulation.